Let's have a look at DNA hybridisation in this very short tutorial. A hybrid of anything is a mixture or a joining of two different things together. That's what a hybrid is. So when we're talking about DNA hybridisation, we're really talking about the joining or mixing of two sources of DNA. And just remember, DNA is universal. By universal, remember, we mean that DNA is DNA. There's no such thing as a human kind of DNA or a fly kind of DNA or a horse kind of DNA or a wheat kind of DNA. DNA has the same molecular structure in any organism on planet Earth. What makes one kind of DNA different from another is simply its base sequence, that is the genes that it has on it. But that's only determined by the genes, the bases that are present in their sequence. So really, the DNA you take out of a human being has exactly the same kind of molecular structure as the DNA you take out of a pine tree. And therefore, theoretically, they could be mixed together and potentially hybridise or join. And this is the idea of DNA hybridisation. If we have a look at this diagram here, we can see this principle at work. So here we have DNA from species A, shown in the pink and the blue strands. So imagine that a segment of a particular bit of DNA was cut out, and there are ways of doing this which we'll look at a bit later in the course. We can cut out a segment of DNA, say from a human cell. Now over here we have DNA from, say, a second species, species B, shown in the pink and the yellow. When heat is applied to DNA, gentle heat, the weak hydrogen bonds that join the nitrogen bases down the middle, which you've already studied, break apart and essentially split the two strands of the DNA apart. So the blue strand separates from the red strand, the pink strand from the yellow strand in B. Now remember, if a base finds its complementary pair, in other words, if an A base finds a T base in DNA, it will join, or vice versa. If a T finds an A, they will join. If a C finds a G, they will join, or if a G finds a C, they will join. And this will happen between any DNA that you mix together. So the concept of DNA hybridisation suggests that if you mixed the strands from species A and species B together and cool them down, this will allow the hydrogen bonds to reform. And the bases that are complementary to each other on the two different lots of DNA will rejoin back together again. So it's possible that you will see a number of bases along the blue strand from species A being able to bond with a number of the bases on the yellow strand from species B. And this can be measured. There are techniques for measuring just how many bases can join up in this hybridised DNA. So this is hybridised DNA. And this can be used for all sorts of things, but one of the really useful things that it's used for is for comparing the similarity or differences between two different species. Obviously, the more bases that join together along the length of the DNA, the more similar is the genetic code of the two species. Therefore, the more closely they're related in evolution. The less commonality there is between them, the less bases that join, then the further distance the species are away from each other, and therefore they're not closely related. So you may have heard, for example, that humans share something like, I think it's about 98 or so % of their DNA with chimps. I might have to check that figure, but for our example here, that it makes the point. What they mean by that is that if you took a segment of chimp DNA and a segment of human DNA and split it and rehybridised it, that anything up to about 98% of all the bases will join back together, which shows that we are very, very closely related in evolutionary terms to the chimps. So this is what DNA hybridisation refers to.